Okay. So for you, let's see, Larissa Ramsey, Davis, White, Reed, Cordone, Boyd. Since you're early, when you send me your email today for attendance with all the words, remind me that you were one of the early ones and you'll get a little extra credit for being early. Yeah.
Okay, thank you.
Thank you guys for being early.
Appreciate it.
Yeah, no problem. Hey,
Thank you.
You're welcome. Since you're here early, does anybody have any questions about anything before we get started?
Um, um, I don't think so.
Okay. Well, if you think of something, let me know. I guess I'll do announcements now because everybody, even though not everyone's here because it's early, they can still refer back to the beginning of the video once I post it for announcements. So I want to remind everybody, I guess this isn't a reminder. Probably we'll finish chapter 3 today. And even if we don't, we'll definitely finish it by Friday. So that means what I would like to do is have the exam on Monday. So Friday, I want to start chapter 5, which will not be on the first exam. And then on Monday, I want to have the exam, and it'll be online. So if anybody has any strong objections to doing it Monday instead of Friday, or even Monday instead of next Wednesday, whatever, send me an email. And if there's enough people that feel strongly about it, and you have to tell me why, even if it's a dumb reason, I don't care. You could just say I have bad tests, I have bad luck on Mondays. I don't care. Like I might not change it because of that, but if you have a dumb reason, that's okay. Share it with me. Let me know when you want to take it. But right now, that seems to be the plan. And I'll remind everybody, this part is a reminder, you really should do the study guides because the exam is going to be online. So you're going to log in like you do now. You're going to get an email, and it's going to say, okay, this exam has been posted, and then you'll have until 8:50 to finish it. So if you've done the study guides, you're familiar with answering these questions online. And as far as the feel of the exam is concerned, it's going to look just like the study guides. It's going to be multiple choice questions that you'll find on Google Classroom. So again, I really, really, really recommend doing the study guides between now and Monday or whenever it is we take the exam. And I've been doing this years, and historically there's a strong correlation between the people who do the study guides and the people who get good grades. Like very rarely do I have someone who fails the exam who also did all the study guides. It happens occasionally. And it might be just that maybe they didn't do it. Maybe they had somebody do it for them. I don't know. But anyway, so yeah, do the study guides, please. And you'll get independent work points for it. So anyway, that's that. Let me have a little bit more than 1 minute left before we get started. So does anybody have any other questions? All right. Well, while we wait, then I'm going to go ahead and pull up the PowerPoint. So yeah, exam probably on Monday. Please study for it. Oh yeah, sometime between now and the exam, assuming it'll be Monday or whenever it is, I'd like to have an exam review. It won't be officially during class time. I'll just pick a time where we can meet online, and I'll send out an email to ask you, and you guys can respond and say, I'd like to meet at this time, or I can't meet at that time. And I will have an exam review where I go through every single question on the study guides, all 3 of the study guides, and then that'll help you with the exam. And I'll record it. So even if you can't make the meeting, you can still watch the video. So yes, if you can't, definitely do the study guides, but also, I would highly recommend watching the exam review if you can't attend it. All right, so let me go ahead and present my screen. Let's see here. Oh, and it's freezing up. Please bear with me. There we go. Please excuse all the background noises. My girls are being grumpy. My poor wife is trying to deal with them and keep them quiet while I am teaching from my COVID room. Let's see. All right. It's almost 8. There we go. Should be presenting my screen soon. Okay, you guys should be seeing my screen. Let me get to the proper slide here on the PowerPoint. We're done talking about, here we go. Proteins is what we were talking about. We were talking about this. We talked about how, there we go. It's 8 o'clock. Okay. Talked about how the most important thing about a protein is its shape. Here we go. Okay. So here's where we left off. Anyway, before we officially get started for today, does anybody have any questions about the material or the course in general? All right, really quickly about the lab, because I've seen some labs. I haven't graded them yet, other than the pre-labs. So I wanted to make a correction so you guys have time to fix it. Come on. Sorry, this is frozen up on me. Okay. Anyway, for the lab, and come see me during office hours if you want me to really explain, because right now I just want to give you a brief explanation and then get back to chapter 3. When you guys did those experiments, you were testing for monosaccharides, you were testing for polysaccharides, you were testing for fats, and you were testing for proteins, right? 4 different things and 4 different tests. And for each of those tests, you had a positive control and a negative control, and everything else you were comparing it to that. So for example, for the monosaccharides, you had water, which you knew did not have monosaccharides, and then you had the glucose solution, which you knew was like pure monosaccharides. So you compared everything to that. So if it looked more like the positive control, you would say it has 3 pluses or maybe 2 pluses. And if it looked more like the negative control, you would say it has 0 pluses or maybe 1 plus. So by definition, and you guys need to go back and check this before I grade it, your positive control should always be 3 pluses. The positive control is always the one that you're comparing it to. So by the way these experiments are set up, there is no way that, for example, your egg solution would have 3 pluses and your pepsin solution would have 2 pluses if we're talking about the protein, because the pepsin solution was your positive control. So anyway, I would take questions on that, but I don't have time. So that's the brief thing I wanted to tell you about the lab from this week. And if, like I said, if you have questions, meet me online for office hours, schedule a time to meet me outside of office hours. So here we go. Can someone please confirm that you can hear me and see the PowerPoint?
I can.
Thank you very much. So the first word for attendance, well, I'll go ahead and jump out and start with that, is going to be, here we go. Let's use this word. Specific is the first word for attendance. So anyway, this is the slide we left off on. We were talking about protein shape and how it's very important. And then we were talking about how the strand of amino acids leads to the shape. And one of the examples I was giving last time was this right here. This is a sulfur. This is the sulfur, and we hadn't talked about this in class, and we never will. But I can tell you, basically, those things are attracted to each other. So because of that, what used to be a long straight string of amino acids like that curved up on itself because these 2 things are attracted to each other. And imagine this, each one of these that they're not showing, each one of these has some special side chain, 1 of 20 special side chains that are repelling things and attracting other things, and that's what gives it its shape. Then we also talked about this. We just showed this other example of an amino acid sequence of a lysosome, and excuse me, I can't talk right now. Anyway, so we talked about this. This isn't anything important for the exam. We're just saying, this is the primary structure. This is the strand. This is the polypeptide, the strand of amino acids. And then finally, once it gets folded into its 3-dimensional shape, then it is a protein. And I get it. You might look at this and say, this picture makes no sense, and this picture makes no sense. And that's fine. It doesn't need to make sense to you. All you need to know is, at this level of biology, is that this protein, these polypeptides, they fold up. And once they fold up into their proper shape, they are considered a protein. That being said, if there is a slight change in the sequence of amino acids that I just showed you, then that affects the ability of the protein to function. Because, again, this is very important. This will be on this exam and another exam. A protein's function is all about its shape. And I just described earlier how the sequence dictates the shape. So obviously, if the sequence dictates the shape of the protein, then if you mess with the sequence of amino acids, then you're going to mess with the shape, and that's going to mess it up. So here's a picture that's not from your book, and I'll give you an example. So you can see here, this you should be familiar with a little bit. Here we have this nitrogen has a hydrogen off of it, right? This carbon has an oxygen. And we know that oxygens are slightly negative and hydrogens are slightly positive. So that's a polar bond, or those are polar molecules, and that is a hydrogen bond right there, right? Hydrogen bond there, and here, and here, and here, like all these hydrogen bonds. So basically, without even being an expert, you can just see this protein and see that it's all spiraled up. And I'm looking at this thing, and I can tell the reason it's spiraled up is because there's hydrogen bonds in between these side groups of these amino acids. So if you were to change this, and maybe if you were to get rid of, I don't know, let's see, carbon dioxide, if you were to get rid of one of these situations where something changed and there was no longer a hydrogen bond there, then that whole helix, right, this whole spiral would unravel. And here's another example. Here we have these hydrogens and these oxygens, again, hydrogen bonded to each other. And if you were to change the sequence and something were to happen to disrupt that hydrogen bond, then you would not have this shape. And again, if you mess up the shape, you mess up the function. So that's just an example. You don't need to know anything about the alpha helix or the beta sheet. The point here is that you need to know that if you change the sequence of a protein, if you change the sequence of amino acids, then that's going to change the shape. And if you change the shape, you change the function, because the shape is the most important thing about a protein. Your book does give a specific example that I might ask you about on some other exam, not necessarily this one, but we'll talk about it later in the semester. If there's a substitution in the hemoglobin protein, and a hemoglobin protein is a blood protein that carries oxygen, it causes a disease called sickle cell. So here's an example. Here's the amino acid sequence, meaning it's not the shape. The only reason it's even curved like this is because they wanted to fit it all on the screen where we could see it. And you might as well just picture this as one long straight string, because that's all this picture is showing. So anyway, there's the amino acid sequence. And this is a real life example. This isn't hypothetical. This is a normal amino acid sequence of a hemoglobin protein. Now, down here is the amino acid sequence of a hemoglobin protein that has sickle cell. And you can see the only difference here is this one amino acid. So which one is that? 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Well, maybe I'm looking at it wrong. Anyway, there's one amino acid that's different. And because the one amino acid is different, you can see that this has a slightly different shape. Look at that purple colored peptide or protein versus this one, right? They look slightly different. But because of that slightly different shaped protein, now the whole red blood cells are completely different shaped, right? The normal red blood cell looks a lot different than the sickle red blood cell. All that because you changed the one amino acid. And again, that's just the way it looks. This is a disease. It's not a thing about how you look. There's a lot of bad things that come along with sickle cell disease, and we'll talk about that later in the semester. But again, the point is, a protein is all about its shape. And if you change the sequence of amino acids, you're going to change the shape. And that's almost always a bad thing. Any questions about that? All right, moving forward. Yeah, so this is sort of what I was saying earlier, but in writing, and with a little bit of additional information. So again, if you even have just one difference, it doesn't even have to be a big difference. If you just have one difference, one different amino acid, one as opposed to another, that is enough to cause it to fold into a different shape. And again, if you have a different shape, that's going to have a different function, and that very often can cause diseases. And also, misfolded proteins are associated with many diseases, including severe nervous system disorders, also something called prions, which we're going to talk about shortly. Also, Alzheimer's disease is associated with some brain proteins. And for now, we'll leave it at that, but we'll talk about it later. Nothing on this slide is going to be on the exam. This is sort of like just a follow-up of what we've been talking about. Here's an example of a prion. So in your brain, you have these normal shaped proteins. These prions are misshaped, and they're interesting because once they come in contact, you can see right here, once they come in contact with a regular shaped protein, it actually causes them to misfold. So what happens is you get these big old chunks of misfolded proteins in your head, and then basically it turns your brain into a sponge, which is where this name comes from. bovine, spongiform, encephalopathy. Man, I can't talk today. But anyway, yeah, that's like mad cow disease is an example of a prion disease. Or Kuru is a prion. That's basically mad cow disease for humans, mostly from cannibalism. Fatal weight loss in deer, elk, and moose. Again, it's another prion, but it's a different species. So none of this will be on the exam. If you can look into it if you're interested for independent work. I always think Kuru is interesting because one, it affects humans, and two, it pretty much wouldn't be spread if it wasn't for people eating other people's heads. So, that's slightly interesting.
Can you spell that?
Which one?
Oh, never mind. I
Okay.
see.
Okay. So, good question, though. I think I understand what you were asking, and then you saw it. So, any other questions? All right, let's move forward. And then we'll talk about prions later in the semester, too. So, for now, I'm going to go we obviously went through it pretty quickly. But later, we'll talk more about it. Anyway. So, I keep saying that a protein's shape is very important, right? It's because its function is very, very much determined by its shape. So obviously, we're going to talk a little bit more about it. Its shape is also sensitive to its environment. And let's talk about why. If you change the temperature, if you change the pH, there's also other factors that I won't even get into that can cause it to unravel. And when we talked about in chapter 2, and this is a good throwback, some information you're going to need to know for the exam. I'll remind you. When we talked about hydrogen bonds, which we can see here in this picture, that's what those little dots represent. Remember we talked about the hydrogen bonds between water molecules, and we were saying that it's because of those hydrogen bonds. Oh, that's so horrible. I'm not even trying to draw it. Whatever. It's because of those hydrogen bonds between water molecules that water is able to absorb a lot of heat before it changes temperature, right? It's able to do that because what happens is those water molecules, they move around a little bit. Like you keep applying heat to it, but the water molecules, because they're hydrogen bonded to each other, they can only move so far because they're kind of like holding hands was the analogy I used. So then finally, once you attach enough heat to it, then those bonds break, then those molecules really start moving, and then the heat starts to increase. The point being, what you should think of is when you see a chemical bond, whether it be the weakest, like right here, which is the hydrogen bond, or even this covalent bond, or this covalent bond, this covalent, right? Every single bond you see there, if you apply enough heat, as far as we're concerned, especially for this conversation, if you apply enough heat, which is energy, to any chemical bond, then it is going to break that chemical bond. That's something you should know, basically from chapter 2. Now, going back to this idea of proteins being sensitive to the environment. That's why if you change the temperature too much, then you are going to unravel. You're going to change the shape of a protein. So for example, again, as you need to know from chapter 2, the hydrogen bonds are the weakest bonds. And you can see all these hydrogen bonds here holding this spiral, this helix together. So if you were to add heat to it, then these bonds would just melt away basically, and then this whole thing would unravel, and you just have one long straight strand of amino acids. You would change the shape of the protein, therefore you would change the shape of the function of the protein. And this is why like high temperatures are so dangerous in humans. Like when you're sick and you're taking your temperature, it literally can. The word's denature. You can denature your proteins. This basically means just melt them. All right, so that's temperature. Let's also talk about pH, and then we'll be done talking about the environment. There's other stuff, but that's all we'll talk about. So again, here we have a hydrogen bond. Does anybody know what this red atom is? I don't expect you to know it, but if you did the models in lab last week, you might remember what color red is or what atom red is. Which element?
Oxygen.
Yeah, there's oxygen. And this little white one right here is hydrogen. Great job, by the way, whoever said oxygen. So there we go. Yeah, we have a hydrogen bond, obviously, between an oxygen and hydrogen. And remember, the oxygen is slightly negative. The hydrogen is slightly positive. There's a symbol for slightly, but I just can't do that with this mouse. Anyway, that's slightly negative. That's slightly positive. Now, let me ask you this. If we were to put this thing in something that was very acidic, then what kind of molecules would it be in? Because and this is also a good throwback to chapter 2, something that you're going to need to know when it comes to talking about pH. If something is very acidic, that means it has a lot of a certain thing other than acid. Obviously acid, yes. But what does it have a lot of? And I'll give you a hint. It's one of these two things. It either has a lot of OH minus or a lot of H pluses. If it's very acidic, what does it have a lot of?
H pluses.
Very good. Yes. And the rest of you need to know that too. That's going to be important for the exam. So yeah, if it's very acidic, there's going to be a lot of these H pluses. So this partially negative oxygen is going to be fully attracted to the H pluses, right? The acid, because that H plus is, that's fully positive. These are just partially positive. Likewise, if you put it in something very basic, this hydrogen would be attracted to that fully negative OH minus, right? The point being is if you change the pH too much, then you're going to break these hydrogen bonds again. And again, the whole thing is going to unravel. So yes, protein shapes are sensitive to the environment. Temperature can unravel it. pH can unravel it. And that's what I'm getting at So any questions about that? All right. Here we are to the last bullet point of this chapter. We might even be able to jump into chapter 5 today, because the thing I want to say about nucleic acids before we get into it is there's going to be a whole chapter devoted to nucleic acids later in the semester. And there's basically going to be a whole exam devoted to nucleic acids. Not directly. There'll be one chapter that talks about the stuff we're about to talk about, which is, what are nucleic acids made of and all that. But then there'll be other chapters that talk about how nucleic acids are reproduced and how they're used. But for now, we're just going to talk about the basics of nucleic acids. I'm going to go through it pretty quickly because I'm going to try not to ask too much about them on the exam, because again, you'll be tested on them later in the semester. So here we go, nucleic acids. The second word for attendance will be this one. If you guys can see, I'm not even going to say it because I just want to make sure people are paying attention. And if you're laying on your couch with your eyes closed listening to me, then you can't hear me say the word. So open your eyes and look at the screen. There's the word, the one I just circled. That's going to be your second word for attendance. Anyway, nucleic acids. These things are important because they store information and they provide instructions for building proteins. And we just got finished talking about proteins. And if you remember on Monday, before we started talking about them, we were talking about why they're so important. Basically, they make up over 50% of the cell weight if you were to take all the water out. And they do so much stuff. Proteins are for structure and for energy storage and enzymes, which we're going to talk about later. So yeah, basically, proteins are very important. So all the instructions on how to build every single protein in your body comes from nucleic acids. So obviously nucleic acids are also very important, because without them, we wouldn't have proteins, and therefore we wouldn't have life. Now, there's two basic types of nucleic acids, and this is something you should know for this exam. You should know that there's DNA and RNA. Later in the semester, you'll need to know some more details, like the fact that DNA is called deoxyribonucleic acid and RNA is called ribonucleic acid. But for now, you don't have to worry about that. For now, just know that there's two types of nucleic acids, DNA and RNA. Any questions about that? Okay. So there's another word you need to know if we're going to talk about DNA and RNA and nucleic acids in general, and that word is gene. If you're taking notes, you don't necessarily need to write this down, because we're going to talk a lot about genes later in the semester. So for now, I just want to introduce you to the concept, and I want you to know what the word means when I use it, but I'm not going to test you on it. But a gene is a unit of inheritance that is encoded in a specific stretch of DNA that programs the amino acid sequence of a polypeptide. And let me back up, and this will make more sense. Remember back here, I said nucleic acids, they provide instructions for building proteins. Well, that's what this is. So a gene, it's just instructions on how to build a protein. So each protein has a specific gene, right? So there's one specific gene in your DNA that says, all right, this is how you build that protein. This is how you build this other protein. So, for example, the mRNA vaccine that you get for COVID-19, if it's the mRNA vaccine, it's a gene, basically. It's not DNA, and we'll talk about that later. But it is a gene. It is a set of instructions that says, all right, here's how you build the outside protein from the COVID virus, basically. And then your body builds it, and then your body's like, oh, that shouldn't be here. And then your body knows how to fight it. But anyway, yes, a gene is basically just, it's a set of instructions on how to build a protein. And of course, what I have written down from this bullet point is exactly what your book says, but I'm just giving you the Cliff Notes version, the layman's terms, because when you break it down, it's saying the same thing. It programs the amino acid sequence of a polypeptide. Yeah, it's the same thing, right? Because remember, a polypeptide is a string of amino acids. And once that string of amino acids it folds and forms its final shape, then we consider it a peptide. So again, a gene, it's just a set of instructions on how to build a protein. And yes, it is a unit of inheritance, meaning, if you were to look at all your DNA, you have a set of instructions from your mom on how to build a particular protein. And then you also have a set of instructions from your dad on how to build the same particular protein. So that's what they mean by unit of inheritance. So yes, it's a set of instructions on how to build a protein. And you have 2 sets for each protein, at least 2 sets for each protein, 1 from each parent. Anyway, I keep saying it's a set of instructions on how to build a set of proteins. So then the question might be, well, what language is this? Are this code? Are these instructions in, right? So is it like you open it up, it's in English and says, okay, the first thing you need is this amino acid. Now attach that amino acid. No, it's not in English, right? The instructions are written in a chemical code, and that chemical code has to be translated because you start with the nucleic acid language of DNA, right? And then also RNA, and I'll explain that later in the semester. So you start in that language, which is just, as you're about to learn, a bunch of A's and T's and C's and G's. And then that has to be translated into the protein language, meaning, all right, first we need this amino acid, then we need that amino acid. But again, all this stuff is going to be covered in thorough detail later in the semester. So for now, if anything for the exam, and probably not even this, but if anything, just know that a gene, it's a set of instructions on how to build a polypeptide or how to build a protein. Are any questions about that? Okay, again, everything that's on this slide right now is going to be covered in detail later in the semester. So I see no need to bother you with it too much right now. Here we go. Here's a picture of everything I just said. Imagine this DNA strand. I mean, it would just go on and on and on. This is just a cut, they just zoomed into this DNA strand. And we looked at this one particular gene. And what happens is there's some things in your cell that reads that gene. And from that, it basically copies it down. But instead of making another strand of DNA, it makes a strand of RNA. And then something else in your cells reads that code and says, okay, this part of that code says bring this amino acid. And then this part of that code says bring this amino acid. That's all it is. It's just the code that says, I'm just going to make up some letters here. Let's see. That one says, I don't know, a T and a Q. And I am really making letters up. And an S and a P, right? That's all it is. So the codes on this RNA say, hey, I need you to bring me a T amino acid. Next, bring a Q amino acid. Next, bring a S amino acid or a P, whatever it is, or a star amino acid. Doesn't matter. The point is, that's what the code is. The code tells you which amino acid to bring to attach to the next one. And again, that won't be on this exam. We'll cover that later. Here's something you should know for the exam, for this exam and the one that we're going to cover later. Because I told you at the beginning of chapter 3, one of the common things you need to know when we talk about all the biological molecules is you need to know the monomers and you need to know the polymers. So the same way that a monosaccharide is the monomer for a carbohydrate and the polysaccharide is the polymer for a carbohydrate, and amino acid is the monomer for a protein, polypeptide is the polymer for a protein. Here we are at nucleic acids. What are the monomers? The monomers are nucleotides. So you need to know that. What are the monomers for nucleotides? They are nucleotides. The monomers for nucleic acids are nucleotides. And just in case you don't remember what I said when we talked about monomers and polymers, or if you forgot or didn't hear it to begin with, remember a polymer is basically a really long molecule that's made up of smaller molecules. So in this case, nucleic acids are made up of a bunch of nucleotides. And like I said earlier, proteins are made up of a bunch of different amino acids, and carbohydrates are made up of a bunch of different monosaccharides. So yes, for the exam, know that the nucleotides are the monomer. Excuse me. Also, and this is kind of similar to protein. Remember with protein, not only did you need to know that the monomer was the amino acid, but you needed to know the 4 parts of the amino acid, right? You needed to know the carbon was bonded to a carboxyl group and an amino group and a hydrogen and a side chain, right? You needed to know those 4 parts of the amino acid. Likewise, for nucleic acids, you need to know the nucleotides have 3 parts. So know these 3 parts. There's a sugar, a phosphate, and a base. And I put those in bold because as far as memorization is concerned, as far as studying, you can just remember sugar, phosphate, and base. Yes, technically, it's a 5-carbon sugar, and here's what it looks like right here. And yes, technically, it's a phosphate group, and technically, it's a nitrogen-containing base. But as far as we're concerned, especially this early in the semester, in this 100-level non-biology major biology class, you will be fine just memorizing sugar, phosphate, and base. So those are the 3 parts of a nucleotide: a sugar, a phosphate, and a base. So any questions about that? Okay, another throwback. To remind you, when we were talking about amino acids, again, you had to know the 4 things that were attached to that carbon, right? The 4 parts of the amino acid. And then I even said you needed to know which part of the amino acid varied, because remember, there's 20 different types of amino acids, and they all have a carbon that's bonded to a hydrogen and an amino group and a carboxyl group. And the only thing that was different for each amino acid was the side chain. And it was the side chain that defined the amino acid, right? Because that was the only part that was different. Likewise, when we're talking about nucleotides, again, you need to know the 3 parts, and you need to know that it is the base that basically defines the nucleotide. It is the base is the only part that's different. So the sugar and the phosphate, if we're talking about DNA, for example, the sugar and the phosphate are always going to be exactly the same. But there's 4 different types of DNA, and that'll be based off of what type of base it has. So any questions about that? All right. Here's a picture of what we're talking about. Let me put a little line here. These are 2 different pictures showing the same thing. So here we have a sugar right here, phosphate right here, and the base, right? And this is the kind of picture that you would have to draw in lab, right? For lab 2, when you had to draw draw molecules. It's a lot like that, right? And then here's a simpler way of looking at it. Same thing though: sugar, phosphate, base. So there we go. Again, for the exam, just know the 3 parts of a nucleotide are a sugar and a phosphate and a base, and then the base is the only part that varies. Any questions about this slide? All right, really quickly before I move forward, like I said earlier, there's 4 different types. Again, let me throw it back before I say this. When we're talking about amino acids, right, when we're talking about proteins, there's 20 different types of amino acids, right? There's a large variety. When we're talking about DNA, there's only 4 types of bases. 4 types of bases. So only 4 types of nucleotides. There's A's, G's, C's, or T's. For now, you don't need to know that. For now, I'm not going to ask you to memorize the 4 types. I might not even ask you the fact that there are 4 types, because again, we're going to cover that later in the semester. Just know that again, the nucleotides are sugars, phosphates, and bases, and that it's the base. That's the part that varies. That's the part that defines the nucleotide. Any questions? All right. So here's everything I said again on a slide. DNA, it's going to be each DNA nucleotide would be one of any of these 4 bases: adenine, guanine, thymine, cytosine, abbreviated A, G, T, or C. Again, you don't need to know that for this exam. You definitely don't need to know the full word. If anything, there will be the letters A, G, T, or C, because we'll talk about those later in the semester. Here's what they look like. There's your A, your G, your T, and your C. Notice that the A's and the G's are double rings. Your T's and your C's are single rings. That'll be important later in the semester. For now, it's not. And here's a picture of what DNA looks like. I'm sure you've seen it before. So here's something that may blow your mind. When you're bringing these nucleotides together, these monomers, when you're bringing these monomers together to make the polymer, guess what? It's a dehydration reaction. So there we go. Here we are at the last chunk of the chapter. And like I promised you, every time we bring something together in this chapter, we call it a dehydration reaction. So at this point, please don't miss that question, right? When you bring things together, it's a dehydration reaction. If you break it apart, it's hydrolysis. Well, here's another hint for the exam. The only reactions we've talked about are dehydration and hydrolysis. So at the very least, if you can't remember which is which, only guess those two. But everything else I'm going to put in there for the multiple choice exam is just a bunch of crap meant to confuse you and meant to trick you. So if you see a word and any question that's asking about a reaction or what kind of reaction it is, if it's not dehydration or hydrolysis, then it's garbage and you'll probably lose more points for guessing it. There's a little hint for you. Anyway, so again, if we're bringing these monomers together, these nucleotides together, that forms the polymer. And here we go. Let's make that the next word for attendance. And a very important word, polynucleotide. So again, the monomer, let me put a big M over here. The monomer for a nucleic acid is the nucleotide. The polymer for a nucleic acid is the polynucleotide. It's very easy. It's in the name. Sort of like the carbohydrates, right? It's in the name. You have a monosaccharide and a polysaccharide. So very similar. Anyway, your book also gives the details like, okay, where exactly are these hydrogen bonds? You don't need to know that for this first exam, but just so you know, these are covalent bonds that we're talking about, and they are between the sugar of one nucleotide to the phosphate of another. So technically, we get this thing called a sugar phosphate backbone. So when you look at a strand of DNA, it's that zigzaggy part. I'll just show you the picture on here. It's called a sugar phosphate backbone. Basically, what we're saying here is it's the sugar and the phosphate that are doing this covalent bonding. It's these nitrogenous bases that are just hanging off that are not doing the covalent bonding. So those, as far as we're concerned at this point in the conversation, those bases are useless. They're not, but as far as we're concerned at this point in the conversation, they're useless. So yeah, for the exam, what's important here is this portion right here. You need to know that it's dehydration reactions that bring these nucleotides together. You need to know that nucleotides are the monomer for our nucleic acids. And you need to know that polynucleotides are the polymers for nucleic acids. And again, this other stuff here and below, that will be important later in the semester. So it won't hurt you to study it. And especially if you can remember this level of detail, you'll surely remember this bigger information. But anyway, are there any questions about this slide? Okay. DNA is double-stranded. Let's see. Yeah, this is going to be important later, but not so much now. You probably already know it's double-stranded. Technically, the word, and you guys have all seen what DNA looks like. There's a name for that. It's called a double helix. That'll be more important later in the semester. Okay. I will say this, though. So if we have a strand of DNA, and this is not right because it's supposed to be coiled, right? It's supposed to be coiled around each other, but I'm drawing it straight just to make things easy. So if that's one strand of DNA, and here's another strand of DNA, and even though you can't see it, what I've drawn is a bunch of sugar. That's a sugar phosphate backbone. There's a sugar there that's covalently bonded to a phosphate, so on and so forth, right? You can't see it, but that's the sugar phosphate backbone. And from that, you have these nitrogenous bases coming off of it. All right. And then also on the other side, on the other strand, there's another hydrogenous base or nitrogenous base coming off of it. So basically what I'm trying to say here at this level, at this portion in the semester, the only thing you need to know is when you see that picture of DNA, well, first of all, it's always coiled and twisted. It doesn't look like what I'm drawing right now. But when you see a picture of DNA, what you're actually looking at is 2 strands of DNA. You're not looking at one, you're looking at 2. So this one particular strand right here on the left, that is held together by those covalent bonds between the sugar and the phosphate. This particular strand on the right, it's held together by those covalent bonds between the sugar and the phosphate, right? Now, what's holding this strand together with this strand? Those are the hydrogen bonds between the nitrogenous bases. So that'll be more, I'm going to say that again later in the semester because you will need to know that later in the semester. For now, I'm just introducing it to you. But again, it's the sugar, it's a covalent bond that holds, this one DNA strand together. It's a covalent bonds that holds this other DNA strand together. It's hydrogen bonds that keep these 2 DNA strands together, this one attached to this one. And then the reason I like to bring that up too is it's a good time to remind you there's a question on the study guide that people seem to be missing a lot. If you're talking about a water molecule, it is covalent bonds that hold a single water molecule together, right? This oxygen is covalently bonded to that hydrogen, and it's also covalently bonded to that hydrogen. So it is covalent bonds that keep this one single water molecule held together. Now, if I were to ask you what's holding these 2 water molecules together, right? The water molecule on the top and the water molecule on the bottom. I'm no longer talking about one water molecule. I'm talking about what's keeping these adjacent water molecules together. And then the answer is hydrogen bonds. That hydrogen is attracted to that oxygen. So sorry, it was just a good time to remind you of that. Anyway, any questions about the DNA or, for that matter, throwing it back to chapter 2 and the water molecules? All right, so for the exam, again, everything on here about DNA is going to be important later in the semester. The fact that A's and T's and C's and G's go together. But for now, you don't need to worry about it. It will be on the study guide, but it won't be on the exam. Here's another picture of what we're talking about. Again, you can see how that DNA, now that you know, if you never noticed before, that's a strand, right, on the left, and that's a strand on the right. So technically, DNA, that's 2 strands, right? There's 2 strands of DNA, and they're held together by these hydrogen bonds. One strand is held to the other strand by hydrogen bonds. But again, each strand, this strand on the right, that's a covalent bond between the sugar and the phosphate. Covalent bond, covalent bond. So any questions about that? All right. Another thing you need to know is that there's some similarities and there's some differences between DNA and RNA. So we're just talking about nucleic acids. In the same way, let me see. I won't say this without confusing anybody. No, I won't even say it. Forget it. So with nucleic acids, yeah, there's 2 different types. There's DNA and there's RNA. And you need to know how they're different and how they're same. They're both polymers or nucleotides. So the monomer is still going to be nucleotide. The polymer is still going to be polynucleotide. That part's the same. The nucleotides still have the same 3 parts, basically, which are sugar, phosphate, and base. So those are the parts that are the same. You need to know that. Are there any questions about that? All right, let's talk about the differences. And the differences for the RNA, it's a different kind of sugar, hence the name. It's called ribonucleic acid, as opposed to deoxyribose nucleic acid. You don't need to know that kind of detail. If anything, just know that one of the 3 differences is, I'll just circle the word sugar. They have different sugars. So yeah, the nucleotides are the same in that they're sugar, phosphate, and base. However, they have different versions of the sugar. Likewise, another difference they have, while we're on the subject of the parts that make up the monomer, DNA has a T, right, as one of its 4 bases. It has a T. Meanwhile, RNA does not have a T as one of its bases. It does not have that option. RNA has a U instead. So yes, again, they have the same 3 basic parts of the nucleotide, which is sugar, phosphate, and base, except I'm telling you now that RNA has a different sugar and also they have different bases. And then finally, the other big difference that you need to know for the exam is RNA is single-stranded and DNA is double-stranded. So are there any questions about what's on the slide? And again, if you're studying for the exam, you don't need to know that RNA is ribose and DNA is deoxyribose. You just need to know they have different sugars. And for now, you don't need to know that RNA, well, most likely you won't need to know that RNA has a U instead of a T. At the very least know that they just have, there is a different base for one over the other. But, it's possible I might ask you that the RNA has a U instead of a T, but we'll see. And again, the RNA is single-stranded, DNA is double-stranded. So no questions about that? Okay, let's see. Okay, yeah, here's just another picture. This is a picture of RNA, and you would hopefully recognize this and say, hey, that looks a lot like DNA. And it does, because to the untrained eye, it's basically the same. The only thing different is this sugar is slightly different. And we won't even talk about why. And then here's that U, right? This base is a U instead of a T. So that's it. All right. So at the end of every chapter, there's this thing called the process of science, where not at the end of the chapter, but in the chapter, there's the thing called the process of science. I always highly recommend reading it. It's great for mental exercises. You will probably need to know it for the study guide. You definitely do not need to know it for the exam. And because you don't need to know it for the exam, we are going to skip it. About lactose intolerance, blah, 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 blah. There we go. Basically, they found out that it is actually slightly interesting. Okay, so remember, a gene is instructions on how to build a protein. Well, the lactase gene is a set of instructions on how to build a protein called lactase, which we learned about in chapter 2. That is an enzyme that helps you break down lactose. So what they found out, we already know from chapter 2 that people who don't have people who are lactose intolerant, they don't have lactase, right? They don't have that enzyme to break down the lactose. So you would assume people who don't have that gene, that protein, must not have the gene. Turns out that's not even the case. Turns out that people who don't have that lactase protein, they do have the gene. It's just that there's one mutation really far away from that gene that switches a C or a T, and that one mutation actually affects the gene really far away. And it's a good prelude, because later in the semester when we talk about genetics, hopefully that'll be more interesting. You'd be like, well, how does that work? How is it that something so far away affected a gene? But yeah, anyway, there it is. So because of this one mutation, even though people have the gene, it's not turned on, basically, that's a good way to put it. So even though they have this set of instructions on how to build the lactase protein, those set of instructions are never turned on, so the protein is never built. Okay, at the end of each chapter, we always have this thing called the evolution connection. And just like the same situation with the process of science, it'll never be on the exam. It'll probably be on the study guide. But because it's not on the exam, I'm going to skip it. It's interesting, though. I highly recommend reading it. If you have any questions, let me know. And that's actually it for chapter 3. 7 minutes. All right. So let's do this. We don't have time to jump into chapter 5. But while I have you guys here, and before I give you the last word for attendance, let's look at this. If you go to classwork on Google Classroom, you can see right here, chapters 1, 2, and 3, right? That's what the exam is going to cover, chapters 1, 2, and 3. You can click on those and skip to the chapters, or you can scroll down. Chapter 1, right here, chapter 1 study guide. That's it. That's where you find it. Click it and do it, because that is going to be these questions that are on the study guide. I'm going to take them. I'm going to reword them. And those are the questions that are going to be on the exam. Obviously, there's only going to be about 50 questions on the exam, so I'm not going to use every single question on every single study guide. But those are the questions that are going to the questions on the study guide, or where the questions on the exam are going to come from, for the most part. And remember, I keep telling you guys to read the syllabus, and I think only 2 of you have. There will be questions from the syllabus on the study or on the exam too. So anyway, there's chapter 1 study guide. Down here, Chapter 2 study guide. Down here, Chapter 3 study guide. So that's how you find it. Also, if you go to the stream and you look at all the posts I post, just go down them chronologically, right? Here's the most recent one. Here's the second to most recent announcement. The third one, fourth one. So when you guys have any questions, if you feel like you're lost and you're out of sorts and you don't know what you're missing and you don't know what you need to do next, just go back to this. Go back to the stream and go back to like the first post of the semester. And that'll be the hardest part, is finding the first one in the semester and make sure you don't go back to the last semester. So if it's in January, you're good. And yeah, just go through those. Here we go. This is the first one. So just go through these one at a time and you can see what you need. And there's links for everything like this. All right, make sure you watch this video and answer these questions. Here's where you do your independent work for week 1. Here's a way you can get in independent work points. It's all here. So go through the stream. Again, if you're feeling lost and you're feeling behind, go through the stream, go through each of my announcements, and please pay attention. And I'm not trying to single anybody out here, but somebody asked me, like, oh, I need to be doing the independent work now. And yes, like I kept repeating, I stopped repeating myself, but for the first, I think, week or two, I kept saying, make sure you guys work on independent work. Make sure you guys work on independent work. Plus, you guys keep getting reminders through the stream to do your independent work. And then someone asked me if they needed to be doing it. So please pay attention. Please, please, please. Anyway, the last word, well, I plan on this being the last word. The last word for attendance is going to be calculator. So we still have 4 minutes. Does anybody have any questions while we're recording about anything? And this would be a good time. And don't feel dumb. Even if you don't have a question, but you know your classmates do, and maybe you think they're afraid to ask, you could just step up and ask for me so I can answer it and get it recorded.
If we wanted to do more than one independent work, will we put it on the same page, like just below the article that we just done one on?
Yes, great question. So each week, as long as it's, so for week 3, we're working on with this sheet. Yeah, so this is the sheet you're working with this week. You can do as many as you want on that sheet.
Okay, thank you.
Yeah, great question. And really, I recommend it, especially, and I said this at the beginning of the semester, and you don't have to if you don't want, because everybody's brain works differently. But for me, I have sort of a short attention span. So I would prefer to do a lot of short papers as opposed to fewer long papers. Like I would just love to answer a little question, write 3 sentences. I was wondering why this, and I looked it up and that, that's the kind of stuff I would like to do. So I recommend writing more than one per week. And you almost need to get the minimum points. And on that note.
Wait, I got a question.
Yes.
So you can just write the papers on anything?
As long as it's about biology, yes. And as long as you use the source.
What you mean by source?
Like if I were to say, I was wondering how many people died from COVID last year. And I looked it up and the answer is 5 million. That wouldn't be good. You would have to say, I was wondering how many people died from COVID last year. And according to this website, and that's your source, it was 5 million.
Oh.
So so you just have to include your source, as in when you look something up, you need to include where you got your information from.
Okay.
Another thing too, and someone's made this mistake, and I don't blame them. It's easy to do. What you guys need to do is make sure you do not reword an article. And I've given you an example. So if you open up your independent worksheet, here's a great example. You can just read it. And what I provided you with is the original. Here's an actual article I found up. Temporary reduction in daily global CO2 emissions during the COVID-19 forced confinement. So you can read this. This is the actual article. And then I gave you an example of what not to do. So that's what this is. You cannot reword something. Like you can't just take an article and then put it in your own words. What you need to do is read it as many times as you need to, and then without looking back at it, just write down what you remember. So please do not reword stuff. And what I mean by reword is, and I'll just do this one sentence as an example, right? This starts off saying, government policies during the COVID-19 pandemic have drastically altered patterns of energy demand around the world. And then I wrote, policies from governments from the COVID-19 pandemic have been very much changed the patterns of the demands of energy worldwide. See, all I did was take the same sentence they wrote and I just changed the words, and that is plagiarism. So be careful with that. Any other questions? You guys, I'm so glad you guys are asking these questions, especially since they're being recorded. All right. So again, at this point, then, unless I hear otherwise strongly and a lot of numbers from you guys, that the exam is going to be on Monday. So Friday, we will start chapter 5, which is not going to be on the first exam. Monday, we will have the exam online. So you'll just look at your emails online. And once the exam posts, you'll find an email. It'll probably be before 8. So you can start as early as you want, but you have to be done by 8:50. And that's it. Sometime between now and then, we'll have a study session where I go over the study guides. And that's it. So no questions? All right. I'll see you all either during office hours or on Friday. And I can't make it to the lab, but if you have questions about the lab, meet me during office hours or find a different time to meet me.
All right. Have a good day.
See ya.